going to make this quick, so.
Yeah, please. I'll do it very quick too.
Okay. Okay, I'm going to get started. I guess we should start. So, hello, everybody. I'm Ned Callan, and Richie Davidson, my good friend and colleague, is with me today. We're going to be brief this morning because we're pretty not limited, but we're structured with time. I want to welcome everybody to the Health Emotions and Wisconsin Symposium on Motion meeting that we have annually each spring. It's a rite of passage. We've got a very exciting lineup today. It's related to translational neuroscience and mental health. This is a meeting that we've been doing for actually 26 years in a row. Richie and I have co-hosted it. It's been a wonderful, wonderful meeting for all of us. And we're thrilled to have all of you with us today, even though it's virtually. Richie, do you want to say a few words about the meeting?
Sure. This is a meeting at which we have our trainees at Wisconsin who are part of the T32 that we've had for more than 30 years, an NIMH-supported training program on emotion research, and the trainees will help to lead the Q&A after each speaker. And so we're thrilled to be able to do this. Actually, last year was the only year we had a lapse in our 26 years. We're happy to be here virtually, and we look forward to hopefully being in person next year. But we have a great lineup today.
Thank you, Richie. I'm going to just run through sort of the schedule a bit. We've condensed the meeting down to one day. It's usually a day and a half. We have outstanding world-class speakers. The focus is on translational neuroscience as it relates to understanding more about psychiatric illnesses and psychiatric symptoms. We're going to start off with Guoping Feng from MIT, followed by Tom Cash, who's going to talk about extended amygdala. We're excited to have David Lewis with us, who is the chair of Psychiatry at Pittsburgh, and is going to talk about his work in relation to understanding the underlying pathophysiology of schizophrenia. Tracy Bale is a real treat to have with us, and we'll be talking about her work related to sex differences as well as intergenerational transmission of psychopathology and pathophysiology. And we're going to finish with Josh Gordon, the director of NIMH, who will talk about his exciting work related to circuitry and psychopathology as well. We have a poster session at the end of the day, and we're going to have a time where everybody's going to come together as well around a discussion that will be at the end of the day. So we hope that you'll stick with us and enjoy the day along with us as we do. I want to particularly thank Amber Sebastian. Amber has been the person that has organized this meeting. Amber, if you'd say just a few words about some of the structural aspects that you'd like people to know about, we'd appreciate it.
All right. Yeah. Hi, everyone. I just want to direct everyone to healthemotions.org. That website has all the program details, including papers from today's speakers. Your profile, if you haven't, there's a top right icon. You can select your time zone, update your camera, add any personal details in there to connect with you. You're going to notice on the right side of the timeline, there is a meeting hub. You can go there anytime during the day today to connect with other attendees, request to interact with them with live chats or calls or exchanging of notes. There's also the poster gallery is up. You can go scope out the posters that are there anytime throughout the day today. Just click on that resource gallery. You also want to attend the virtual poster session, 4:30 Central. Just click view presentation and you'll be able to interact with poster authors and listen to their presentations. Also, while navigating this virtual platform, you can always return little red return to timeline in the upper right. We also invite you to ask questions throughout the day today. There's a Q&A box. Ask those questions. Ned and Richie will be monitoring this. And if we don't have time to get to your question during the post-doc talk discussion, please join during the general discussion with all presenters. If you encounter any technical issues at all today, especially if you were presenting a poster or something like that, there is a live support icon at the top right. So please do click on that if you want to reach out to the support staff on the back end. You can also watch, if you want to learn more about the history of the symposium, there's a history of symposium video available at the top of the timeline. You can watch any time during the day today. It's about 10 minutes. All of today's talks will be recorded and made available for everyone afterwards. So stay tuned for an email in the coming few days. So
thank you, Amber. I also want to gratefully acknowledge our supporters, the Healthy Minds Institute, which Richie heads up, the Health Emotions Research Institute, part of the Department of Psychiatry, which Richie and I founded initially, the Department of Psychiatry, and Therapeutics for their generous support, which they've been supplying to us over many years, and to thank Joe Belanoff in particular for that. So what I'd like to do now is introduce our first speaker. As I mentioned, the focus today is related to translational neuroscience and as it relates to psychiatry, and there's nobody better to lead us off in this endeavor than Guoping Feng from MIT University. Guoping has done groundbreaking work related to molecular genetics as it relates to circuits and behavior. His particular work in relation to autism and autism-like behaviors, as I mentioned, is groundbreaking. He's translated this work not only in elegant models in rodents, but to non-human primates as well, which is particularly exciting from my perspective. We're thrilled to have him with us. If you want to learn more, which I'm sure you do, his bio is posted, so I'm not going to go through all the details, but just suffice it to say that it's just a wonderful pleasure to have him with us today. So I'm not going to take up any more of his time and want to introduce Guoping. Thank you so much, Guoping, for being with us.
Thank you. Can you see now? Okay. Can you see now?
Yeah, we can see. Yes, okay, we can
great.
see.
Okay, great. Thank you, Ned and Richie, for inviting me. This is an exciting symposium. Thank you for organizing such a wonderful symposium. So today I'm going to talk about some of the work we have been doing and try to, mostly I'm looking forward to also to the discussions, the brainstorm, how can we work together to move the field forward and really think hard on what's next thing we need to do. So so my lab, like many other labs, we are really interested in understanding the development, function, and dysfunction of synapse. In particularly, my lab is interested in how dysfunction of synapse can lead to specific circuit defects that cause abnormal behaviors related to neurodevelopment and psychiatric disorders. So recently, I'm going to focus mostly on ASDs, and mostly recently large-scale genetic studies have identified the synaptic dysfunction as a key mechanism for ASD. And this I'm borrowing slides from Thomas Bourgeon's review and showing that these brown labeled proteins at synapses are strongly linked or implicated in autism spectrum disorders. So in my lab, like many other labs, we are really interested in focusing on the postsynaptic development. And one of our key focuses on scaffolding proteins. Scaffolding proteins at the excited synapses are critical for the assembly and the dynamics and modulation of synaptic functions at the synapse. Well, today I'm going to use one of these scaffolds called PS95 SAPAP Shank, in particular Shank proteins in synaptic function dysfunction. This work has been, built on many previous works, especially Mary Kennedy, Morgan Shan, Richard Hugener, Tobias Boger in Germany, and David Brad at the time was at the UCSF. So we want to understand one of the particular proteins called Shank. Shank is the scaffolding protein linking the scaffold PS95 SAPAP to the cytoskeleton through their interaction with the actin binding proteins. So the reason we are interested in Shank3, one of the Shanks, there are three Shanks, like Shanks 1, 2, 3. We are particularly interested in Shank3 because Shank3 mutations are strongly linked to autism spectrum disorders. This was originally identified called Phelan-McDermott syndrome, which is a deletion of chromosome 22q13.3. and which include the Shank3 gene. And later on, a study from Thomas, it's called Phelan-McDermott syndrome because it was originally identified by Dr. Phelan and Dr. McDermott. But later on, Thomas Bourgeois and many other works show that genetic mutation in Shank3 is most most likely the cause of the neurodevelopmental disorder phenotypes and pathology. So later on, the larger scale genetic sequencing study also identified many Shank3 mutations, including loss of function mutation, small deletions in autism spectrum disorder. So now it's very clear like Shank3 mutation is a monogenic cause of severe neurodevelopmental disorder, and it has many different phenotypes, including hypertonia, developmental delays, very severe intellectual disability. IQ on average is around 30. And also severe impaired in language development. And of course, it has social interaction deficit of the Joe Buxbaum study from Mount Sinai showed that about 84% of the patients can be diagnosed as autism or autism spectrum disorders. They also have many other comorbid conditions, including sleep disorder, seizure, about 30% of people, and sensory abnormalities. So we are trying to use animal models to try to understand what are the neurobiological basis of these issues. So as I mentioned, there are 3 Shanks. Shank3 caught our attention is because Shank3 is the only Shank highly expressed in the striatum and the thalamus. So my lab is particularly interested in the cortical striatal thalamocortical circuits. And the reason is, first of all, this circuit is relatively conserved evolutionarily. When we use animal models, we want to use we want to study a system that can hopefully help us not only understand the mouse, but understand the human conditions. The second reason is that dysfunctions circuit from clinical and animal studies have been linked to many psychiatric neurodevelopmental disorders, including ASD, OCD, ADHD, Tourette's, and schizophrenia. So they are involved in habit formation, repetitive behaviors, but also sensory processing, attention, sleep, cognitive inflexibility, and cognition. So that give us an opportunity to really dissect in how Shank3 mutation may lead to a variety of different phenotypes. So we actually did the first we did was the knockout. Just straight knockout. I'm going through very briefly. Indeed, in the striatum, use both biochemical, electrophysiology, and morphological studies. These might have the mutation or deletion of Shank3 lead to biochemical defect, molecular defect at the synapses, including loss of or reduction of other scaffolding proteins, including PS93 and SAPAP3 and the HOMA, but they also lead to reduction of glutamate receptor subunit like NMDA receptors and AMPA receptors. Also, the electrophysiologically, you see use a whole cell patch clamp. You see mini EPSC, mostly frequency reduction, but also peak current reduction, and also it has reduced the spine. These are the at the pathological level, you can see from behavior level also has many issues with these mice, including repetitive compulsive behavior. They grooming their skin off, and they have social interaction deficit, and they also sensory oversensitivity, and also sleep and locomotion issues. So we're trying to use the mouse model to figure out what circuits are responsible for individual phenotype. Is there a common circuits are common cause of all these different problems. What we found is actually each individual's phenotype has a very different pathology at a cellular level and at a circuit level. So for example, I'm going to go through, not going to show you a lot of details. I want to show some of the details later on more recent studies. We show that the defect in the striatal medium indirect pathway medium spinal neuron, reduction of that activity actually is responsible for the repetitive behavior. So if we use a DRAS developed by Brian Ross at UNC to slightly increase the activity of indirect pathway medium spinal neuron, you can actually correct this repetitive behavior, suggesting that the pathology is indeed, as many other people have shown, including my colleagues at MIT and Graybill, that the striatum play a very critical role in the repetitive behavior. So Then besides this repetitive behavior, they grooming the skin off, they also have many other behavior defects. I'm going to show you a video to show how they interact socially. So on the video, on the screen, you will see two cages. One is on the left is the wild type. Two wild type mice. We basically drop two mice, stranger mouse into the cage, and on the right are two mutant mice. What you will see is that in the mutant mice, they bump into each other. They will not, interact. And the mutant wild type mice, it's very normal. They will go find the other mouse, sniff the other mouse behind. That's their social. That you can see. Then they will, cuddle together. Mutant mice, they will, get away as far away as possible. This is what you can see whether this is related to human social. It's hard to say, but it's a very interesting biological behavior, right? So what causes this kind of a single gene mutation causes such a complex behavior defect? So my lab and my former postdoc lab, actually, he now is a professor in China, continued this line work. It showed that the reduced neuronal activity, in this case, in pyramidal neurons, in the anterior cingulate cortex, actually is the cause of this, at least the partial cause of this defect. If you increase the neuronal activity in pyramidal neuron in the ACC, you can actually rescue social interaction deficit. So this is a second phenotype in a different, now the defect is in a different brain region and a different cell type. So besides this, these mice also have a very interesting phenotype we call phobia of novel objects. We think it's a sensory oversensitivity. So I'm showing a video to show that there are phenotypes. So in this video, right side is Y type, 3 Y type mice. On the left side are 2 mutant mice. What we did is just drop a plastic ball in the middle of their cage, just place in the middle cage, and mice are usually interested in explore new object. So what you will see is Y type mice will approach it and explore. Pretty soon it will get on top of it. The mutant mice, you can see when their head getting close, they will startle back. It will suddenly retract. So what we think is maybe whiskers just touch it and very sensitive. So we thought this might be a good model to study sensory oversensitivity, which is a major issue for many of the neurodevelopmental disorders, including ASD patients. So you can see these Y type they go to get on top of. So we work together with Chris Moore's lab at Brown University, and he's the expert in somatic sensory systems. So we established the whisk assay. Basically, you use piezo stimulation. You can control the strength of stimulation to see, and then train mice for basically for reward assay. To if they sense it, they will correct it. They will get a water reward. So what we found is that actually the mutant mice learns much faster because they can sense the very weak stimulus that Y type couldn't sense. They already can sense it, so they learn actually very quickly. So that means their sensitivity, you can see at a maximum sensitivity, if you give a maximum at a maximum stimulation, they are same. But at a weak stimulation, the mutant mice are much more sensitive. They can detect it. So they're suggesting this maybe this whisk withdrawal we saw, novel object phobia is the reason for that is maybe they have a sensory hypersensitivity. So we want to understand what is the cause of this. So we use calcium imaging, and Chen, another postdoc, and with Chris Diaz, who is Chris Moore's postdoc, work together, look at the calcium imaging in the somatosensory cortex. So we imaged both pyramidal neurons and inhibitor neurons. What we found is surprisingly in these somatosensory cortex of mutant mice, actually pyramidal neural activity is increased. Although Shank3 is critical for excited synapse function, actually in these mice in the somatosensory cortex, actually their pyramidal neural activity actually increased. What we then look at the inhibitor neuron. What we found is that the inhibitor neuron activity in the somatosensory cortex is dramatically reduced. So what we think is that in this case, the cause is probably coming from the defect in the inhibitor neuron. Although the Shank3 is knockout from germline in everywhere, right? So, but for reasons it's still not clear what, but it seems like inhibited neuron defect maybe caused the increased activity in excited neuron that caused the hypersensitivity. So to test that, we actually made a conditional knockout. We only knock out used by Cree injection, knock out Shank3 in the inhibited neuron. And we found exactly the same phenomena. We found excited neuron, in this case, is normal with normal levels of Shank3, have hyperactivity, and we found a similar behavior. So we think this, if we can correct in the future just the inhibited neuron defect in the somatosensory cortex, we may be able to rescue the sensory defects. That's the experiment is still ongoing. So I tell you all this is because it's bothered us a lot, because all of us try to understand how can we develop therapeutics for autism spectrum disorders. But for this, from one animal model, this is only a single monogenic AS model. We already found a neurobiological defect in multiple cell types and in multiple circuits linked to various behavior abnormalities. So there are hundreds of causative genes or risk genes. So how can we develop therapeutics? So that things bothered us for a long time. So from my lab, many other labs study on Shank3 and other gene monogenic disorders found a similar thing. There are many different defects in different cell types, in different molecules, and lead to a variety of different behavior phenotypes. So we think that for that we adjust our strategy. We think there are three main strategies in the future where we can work on, at least three, and there are probably many more. So we are focusing on three things. We think that for some of the monogenic disorders, we can develop gene therapy with the current technology that can correct the genetic defect. Is probably the has the most potential, because many of the genes are monogenic mutations. I mean, mutations on a single gene, but their consequences are actually not monogenic, right? Shank3 affect many other proteins. Fragile X affect hundreds of genes. Rett syndrome affect also hundreds of genes. So all these, if we can correct the genetic mutation at the roots of the cause, then we have a much better chance to treat them or even cure them. But many of them are not monogenic, right? Large majority, current estimation is 80% are not monogenic. So what do we do with them? So we think that with the new technology, we can actually much precisely understand the sex dysfunction, and if we can correct the sex dysfunction, we can probably relieve some of the most debilitating symptoms that will also be extremely helpful for the life of patients. Then the third, which is less studied, I think has a lot of potential, is if we can find very early during development, early on defect, we might be able to prevent the sex malformation or malfunction, because many of our circuits are established by genetic programs, molecular mechanisms. But all the circuits, almost all of them, are modified, refined during our development, during the grow up. Yeah, that's why everyone has different experience. You have different, cognitive functions. You have different ideas. You make different judgment, right? So these are, because the activity-dependent refinement of circuits is a critical part of our development. If there's a defect at the synapse or other functions, then your activity pattern will be different. So refinement will be different. So if we can correct that early event, we might be able in the future to prevent the malformation, which may be much more effective. So that's also something we have been trying to test, whether early intervention would be sufficient at a molecular or cellular level, sufficient to completely prevent the future, for example, sensory oversensitivity. So I'm going to use the example. One of the things for critical is that if we want to do, whether it's gene therapy or whether it's developmental rescue, when it's rescuable, right? It's a development disorder. So each gene probably different. So we specifically tested Shank3. So we made a mouse is a knockout, but allow us to turn the gene back on, use Cree recombinase that allow us to turn whenever we want to turn it on. So it's a tomography inducible Cree line. So what we found is these mice grow up as a knockout, But if we cross these mice to tamoxifen Cre-ER, then we can turn on the Shank3 gene expression by feeding the mice tamoxifen. Here we use TM. So you can see the wild type. This knockout is the inverted exon, which is a knockout. Then if we feed them tamoxifen, you can reintroduce the Shank3 expression, so restore the major isoforms. So what we found is, if the mouse grow up, as a knockout, they have all the phenotypes that I showed you before. Then we start feeding them with tamoxifen, about 3 months old mice. When you turn the Shank3 gene back on at that time, what can you rescue? So what we found is that at a molecular level, it can rescue all the defects. That means once you have the Shank3 protein, all the SAPAP3, HOMA, glutamate receptor subunit, all back to the synapse, to normal. Red is the knockout, or blue is the tamoxifen treated, and the dashed line is the wild type level. You can see they're all correct. What really surprised us, this is all from striatum. What really surprised us was that the spine density is reduced in the knockout, but even in the adult, at least in the striatum, actually you can restore the spine numbers. So that means they are not only molecular in adult, not in the striatum, not only you have a molecular plasticity, you also have structural plasticity. And similarly, functionally, they also rescue. We both use field recordings and use single cell recording, whole cell patch. We can restore the functional synaptic function when you re-express Shank3 in adult mice. And this is recording again in the striatum. So phenotypically, we also found it's interesting that you can actually rescue not only the molecular and the function, you rescue the behavior. So this is before the tamoxifen feeding, as I mentioned, they grow up like a knockout. They have repetitive grooming, they have lesions. They're basically grooming their skin off. But after tamoxifen treatment, the same mouse, you can see not only the lesion heals, their hair grow back, right? The only thing the hair grow back here difference is the white hair. And this is probably have something to do with the treatment of tamoxifen, nothing to do with the Shank3. And also surprising thing to us is we can also rescue the social deficit phenotype. Wild type mice, this is the famous 3-chamber assay modified by Jackie Quarry at the UC Davis, now at the NIH. You can see now at the UC Davis was at the NIH. So what you can see is you put mice in the middle. There are 3 chambers. And right is the small cage, you put an object. Left, you put a stranger mouse. Mice like to go interact with other mouse, but a knockout does not have a preference. And after in adult, if you restore the Shank3 gene expression, you can restore this preference for interacting with the mice. So at least for repetitive behavior and social interaction, if you restore the Shank3 expression in adult, you can actually rescue them. But that's actually not the whole story. We found many other things you cannot rescue in adult, including motor activity in the open field, anxiety-like behavior, motor learning in the rotor rod, and also including the sensory oversensitivity. We call the novel object phobia. So that suggesting that indeed there is a developmental critical window, right? It has been studied in many systems, especially in the visual system by many people, Michael Stryker, Carl Hench, all have shown this. There are critical windows, including language. So if you pass that critical window, many things you cannot rescue anymore. But is there a developmental window, postnatal window we can still manipulate? So we test them at the younger mice. We fed the tamoxifen at a P10 or P21. So at least at a P21, you can actually improve most of including the rotor rod or anxiety-like behavior or motor activity. So suggesting that indeed, at least for the Shank3 mouse model, they are critical circuit-specific. They are circuit-specific adult plasticity, but not everywhere. They are indeed the critical development windows for some circuits and behavior. However, the good news is there is a postnatal window, at least from mouse study, for gene therapy. If we can develop a gene therapy to restore the Shank3 gene expression, there seems to be a postnatal window that allow us to correct the function, not only at a molecular level, probably at the behavior level. So we tried this idea. So we designed Shank3 is too big for AAV packaging, so we made a mini Shank. We collected we think the most important aspect of the Shank3, and this mini gene still goes synapse. So if we tag it with GLP, you can see mini Shank3 GLP goes synapse, matches PS95 very well. So then we use bendamine and Viviana Gradanaro's PHPE B vector, and expressed with systematic injection into the vein, facial vein, then it will spread past the blood-brain barrier, get into the whole brain. It's actually, in mice, it works really well. And we did at a P0, P7, and a P28. What we found is that you can actually express the AAV mini Shank3 very well. Then we tested the behavior around 2 months old. What we found is it can rescue at least P0 and P7, it can rescue every aspect, including synaptic physiology, biochemical PSD proteins, and also behavior, including social behavior, motor, rod, every aspect that we can rescue. So suggesting indeed, there is a postnatal window for gene therapy. Use a mouse model. Whether this can be turned into humans, it's still difficult to say because the mouse development may be very different. So suggesting that, but at least give the hope that this construct that we can actually potentially move into testing more preclinically in other animals than moving into clinic. Actually, this construct has been recently licensed from MIT and Broad by a biotech company. They try to push it into clinical trials. So I'm very excited to see help them to whether this can actually develop into a future therapy for help patients. So then I want to just spend a little bit of time talking about how, since monogenic is still a small portion of the patient, how can we develop a much more precise circuit manipulation for polygenic causes and to release some of the most debilitating symptoms. So the new technology now allows to do much more complicated and much more sophisticated actually circuit dissection, right? So here I'm going to use one of the structures in this cortical striatal circuits called thalamic reticular nucleus. The reason we focus on this thalamic reticular nucleus is because there are two reasons. It's a critical role in actually modulating thalamocortical output, right? So its function as a gatekeeper. Actually, it will select, amplify relevant information from sensory and other domains, and suppress distractions so we can pay attention, we can sleep better, and also TIN dysfunction, which can be measured with as a surrogate of sleep spindle, has been found in many psychiatric disorders, including ASD and schizophrenia. So the other reasons we found that many of the neurodevelopment disorder, autism and schizophrenia genes are highly expressed in this TIN. TIN actually is a thalamic reticular nucleus, is a very thin layer, but a very big and wrapping around the thalamus. It's all GABAergic, and it's all inhibited GABAergic neurons. So in mice, they are all PV positive, actually, PV neurons. And we found that many of the genes are actually highly expressed in this was led to us look at one of the deletions called patch D1. Patch D1 actually deletion is linked to another neurodevelopment disorder. It's not a syndrome. It's not a syndromic. It basically patients show many different symptoms, including developmental delays, sleeping disorder, ADHD, hypertonia, very aggressive, and ASD and intellectual disability. So we want to understand what's the role of TIN in this development disorder. So as I mentioned, get us excited is not only ASD genes, but also schizophrenia genes. Some of high-risk genes are highly expressing the TIN. This is the mouse section. You can see the TIN is wrapped around the thalamus. So we made a patch D1 knockout. What we found is that indeed, if you knock out patch D1 in germline, you see sleep spindle defect, you see TIN bursting defect, and this leaded actually to the actually reduce the sleep, actually disrupt the sleep. So we found that in these mice, their sleep bouts are much shorter. That means they wake up much more frequently. So these mice also have attention deficit. I don't have time to go through the details. This was developed by Mike Halasa's lab to show to test the attention. We found these mice can do tasks really well, but if you give them a distractor, and then they make a lot more mistakes, suggesting they cannot focus as well as the wild-type mice. And in the open field, they are also very hyperactive. So we think this is like a model which is the ADHD-like behavior. But besides these, sleep, attention, and hyperactivity, these mice also have many other problems. That actually amazes me. Actually, models are many of the relevant phenotypes in humans. They have learning disability. They are very aggressive. You actually don't have the genotype. You stick your hand into the cage, you know which one. The one bites you are homozygous, which is the male, right? This is the X chromosome linked to this gene. And they also have motor problem. So then the question is, what is, are all these caused by TRN? Because at the early postnatal days, the PhD is very highly expressed in the TRN. But later on, the expression is still high in the TRN, but in many other brain regions, including striatum and the cortex and the cerebellum. So we want to figure out what is, which part can be attributed to the TRN, which part not. So we made a conditional knockout, just knock out the PhD1 in the TRN. So this is, you can see the wild type, this is the TRN knockout, you can see everywhere else is there, but the TRN expression is gone. What we found in this specific knockout, we only have attention deficit, hyperactivity, and sleep disruption. So consistent with the TRN function, but no, for example, no defect in fear conditioning test, which is the associated learning. They are not aggressive. They have no motor defect. So suggesting that indeed the TRN defect only responsible for a part of the symptoms. And you're probably all very clear that sleep disruption, ADHD, and sensory abnormality are debilitating symptoms. They are common in many neurodevelopmental disorders. Right? So TRN play a critical role in this part. So the question for us is, can we find a more specific circuits in the TRN, or ideally more specific target that we can modulate the function to correct this problem in neurodevelopmental disorders without affecting the whole brain function. Right? So we decided to do a detailed analysis using single cell RNA-seq, and this allows us to dissecting we made a mice PVTD tomato mice, allow us to actually dissecting the TRN out and did a single cell RNA-seq. So what we found is, indeed, in the TRN, there are two groups of neurons. Both are GABAergic, but they actually have different gene expression patterns. They have a gradient expression, and one group we can use a marker gene called SPP1. The other group we use the ECEL1 as a marker gene. You can see they have, these are some of the genes showed here. They express an opposite gradient. So suggesting that there are two groups of cells in the TRN, and so we were very interested in, do they, what's their function? How do they form connections? Do they have sub networks in TRN regulate the different functions of the TRN? So what we found is that indeed, even anatomically, actually, you can tell the difference. What you can find is that the SPP1 neuron is in the middle of the TRN, the red, and the ECEL1 cell, which is the green here within situ, they are actually at the edge. So because they are in the thalamus, there are two major groups of nuclei. One is we call the first order. They basically receive peripheral sensory information, then relay the information to the cortex. The other group we call the higher order, which is basically receive input from the cortex, different regions of the cortex. They output also again to the cortex. Their function more likely is more, regulated multisensory integrations and other higher cognitive functions. TRN actually projected to both, regulated both of these nuclei in the thalamus. The question is, do they come from same type of TRN neurons, or they have segregations? So what we use the retrograde tracing, we found is very interesting. In the TRN, as I mentioned, there are molecular groups, right? So one group, SPP1 cells are actually, if you're recording from them, they are high bursting. The other ECA1 cells are low bursting. But the most interesting to us is that these two group of neurons, as I mentioned in the TRN, they have different locations. SPP1 neurons in the middle, ECA1 on the edge, but they also have a very precise projection. So the SPP1 neuron in the middle of the TRN, they are actually 80% or more projecting to the first order peripheral sensory relay thalamic nuclei. For higher order, like a medial dorsal thalamus, these are thalamic nuclei, they receive the ECA1 neurons projection for TRN. So they have different in vivo connectivity. So you form two separate networks actually in the TRN. So now, use this knowledge, since we know all the genes expressing every of the cells, we actually now have identified not only drug target, but also a compound that can correct attention deficit, sleeping problem, hyperactivity in patch D1. So we got excited. So this might be a very good target that allow us to do this kind of study in the future in clinical study. However, as we all know, there are many challenges we use mouse models, right? So for gene therapy, there are, for example, some of the major issues is how do you get into the brain? How many neurons actually do you need to have the Shank3 restored that have a therapeutic effect? Mouse brain so small, it's much easier actually to have 70 to 80%. Right now, there's no way in bigger brains we can get that level. The PHPEB does not penetrate the blood-brain barrier very well at all in monkeys actually. And no one has tested in human yet. So the other thing is, what is the effect of age? Do we have to treat a 1 year old? Can we treat a 5 years old? All these will have difficult to determine these mouse models. But sex-specific drugs, right? The prefrontal cortex in mouse and the monkey and the humans are very different. And we now many studies from Allen Brain Institute, from Steve McCarroll's group with us together show that actually there are many gene expression differences. There are actually different cell types between the primates, including humans and the rodents. So for all these reasons, many years ago, we start to think about how can we move one step forward for benefit of patients. So we decided to explore the possibility to genetic engineering monkey non-human primate models. So it has been a very long road. It's very difficult, but the CRISPR development of CRISPR actually has really speeded up. So now at MIT and Broad, we have two major projects. One is using marmoset, the other is using macaque. The marmoset of the colonies are all at Broad and MIT. The macaque work actually is a large collaboration with NIH support with our colleagues in China. So I'm going to just mention briefly to show you what's the difference, what's the potential with these models. So we again started with Shank3 mutation. We were very lucky. Our collaborator in China, very experienced. So far, all the macaque monkey knockout has been generated actually in China, and published ones. And so we actually had 5 Shank3 monkeys. We had heterozygous, and by just accident we have the homozygous. So we found that these monkeys are, we can do a lot of studies non-invasively. So for example, we can give them activity watch, right? So like us, we all want to know how many steps I walked and how well did I sleep. And we give them activity watch. You can see the blue is the control. Red M3 is a heterozygous, M2 is a homozygous. You can see that day and night is 12-hour light cycles. You can see in the wild type, the high activity during the day and very low activity during night. They sleep really well, right? But in the heterozygous, you already see, by the way, all patients are heterozygous. So you can see that day activity is reduced and the night activity is significantly increased. So they don't sleep, they have a lot of sleep problems. And this is even worse for the homozygous. So then I just want to say, show you, some videos to show it's easier to see than bar graphs show how they do social interactions. So in this case, we actually probe each control or mutant monkey with 10 Y-type monkeys, stranger monkeys. They never saw it before. Because monkeys, they have personality, they may use one, they may not like other monkeys, and they don't interact, it will really screw up your data. So each monkey is probed with 10 Y-type probe monkey, we call them. So probe monkey has a green color on their neck. You can see very easy. So now they put into 2 cages, and now they are separated. So this person is coming to release the divider. Now suddenly they can interact with each other. So this is 2, Y-type, and you can see now the divider is released, right? So you can see it has a green color. I don't know whether you can see, and so now they can. Yeah, so they follow. So the white, they never saw each other, but they're still interested in each other, engaged, and they back and forth and follow each other. So this is a normal, Y-type monkey behavior. That you see the green color pretty clear now. So they are interacting with each other. The next video is identical probe monkey, except the other monkey is the heterozygous Shank3 mutant monkey. So what you will see is that. So first of all, this mutant monkey has, has repetitive flipping, repetitive behaviors. This green monkey is the same probe monkey, right? So now this person is coming. She's going to release the divider, and so suddenly now they can interact with each other. So the probe monkey goes other side, right? So the mutant monkey goes to the other cage. The mutant monkey is interested in something outside. But if you pay attention, you never actually look at that other monkey, right? So it's always looking at something. And actually, we found that the behavior of the probe monkey pretty soon figured out that monkey is not interested in me, and their behavior is very different. So you can see the very clear differences. Even they are very close by, this mutant monkey don't look at the other monkey. They're very different, right? So they don't follow each other. This probe monkey try to follow, but the other monkey will get away with it. So we think this kind of phenomenon may be more. It's more relevant to human symptoms. So we hope that if we can use this monkey to figure out the circuits and correct the problem, maybe it might be more translational. And so we can also do the function MRIs with these monkeys, and we actually did function MRI with both with the heterozygous and homozygous monkeys. We see very significant differences between the 2 groups. This is our resting state local connectivity. You can see in thalamic striatum, they are dramatically reduced, but in the sensory cortex, whether it's a somatosensory visual cortex, their local connectivity activity is increased. So we're hoping that with this kind of model, it will help us to really better understanding the neurocircuit basis of some sophisticated or high order behaviors like social interactions and communications. And now we have actually a large number of F1 second generations. We also made successful learning from our Chinese colleagues with knockout in macaque. We actually now very successfully made Shank3 mutant marmoset at MIT and abroad. We have also have F1s now, so we are marmoset are very, very social, so we're starting to see what's the difference between these groups. So I'm going to stop here just because of time, and we're hoping that Wisconsin Primate Center has a huge expertise. We're hoping for a lot of collaboration in the future to really explore these models and to see whether, we don't know yet, so see whether this will be a better translation model for brain disorders or not, right? So we need to test them. So finally, I want to acknowledge a lot of people, my lab people who did all this work I mentioned along the way, and some of them. And I want to mention our non-human primate team. It's not my lab project. It's no single lab can do this. So we have a lot of team work together. It's almost like a Boston area, including Broad, MIT, Harvard, Boston area project. I'm just very fortunate to work with all these people. And we also have collaborated from NIH, Jim Pickle, Alfonso Silva, our Japanese colleague is extremely helpful. We send many many batches of people to Japan to learn marmoset technologies and reproduction. Dr. Okano, Dr. Sasaki, and we have many of our Chinese colleagues, especially Zhonghua Lu and Li Pingwang. They really helped to, and Sihua, and really helped to generate the Shank3 macaque monkeys. And finally, I want to thank all the people to support us. It's impossible without our donors for the Tan Yang Center and all the Stanley Center and Tan Yang family and the Stanley family and many of the other supporters, including NIH and IMH and Brain Initiative. Thank you.
Thank you so much, Guoping. It's just beautiful, absolutely beautiful work. And what's so exciting is the capacity to begin to translate this to humans. Just really, really, really wonderful. Thank you. Richie, do you want to go ahead and lead us off on our discussion?
Sure. Thank you so much, Guoping. That was really a masterful presentation. And Justin Russell, who is one of the trainees in our T32 here at Wisconsin, will be leading off the discussion. And these are questions that have been aggregated from several different trainees who have been stimulated by and read your work over the course of this semester. So, Justin.
Thank you, Richie, and thank you, Dr. Feng, for such a brilliant presentation.
Thank you.
So our first question really, and you kind of alluded to this in your talk a bit, but what barriers would you say exist to applying your work to the 60% of individuals with intellectual disability and 80% of individuals with autism spectrum disorders that's not attributable to single rare genetic variants?
Yeah, so this is a really good question, very difficult question. So in my view, so I think, study the monogenic disorder is not only for the monogenic disorder. I think let's think about social deficit, right? So if we, clinically we can exclude the social deficit caused from intellectual disability, I think that whether it's a monogenic disorder and the polygenic disorder, they have the very similar circuit mechanism. That's our hope. And I personally don't think there are two separate circuits for monogenic disorder for social behavior, for social, or for anxiety, or for ADHD. So understanding from the experimental research point of view, we, unlike other studies, for brain disorder, we need models. We cannot just take the brain from patient, right? We don't have that luxury for cancer research. So how do we model them? So we need use the model to, but we cannot right now model polygenic disorder. So monogenic genetic disorders actually give us a hope that we will figure out the circuits and we will test them in other conditions. That's our hope.
Wonderful, thank you. Next, what kinds of phenotyping would you like to see in clinical research to better approximate the cell-specific or circuit-specific functional changes that you observe in animals?
Can you repeat it again?
Sure, I'd be happy to. What kinds of phenotyping would you like to see in clinical research to better approximate the cell-specific or circuit-specific functional changes that you observe in your animals?
So that's a really good question. So we actually, what we want to understand is, we want to see from clinical is what is the most robust phenotype, because clinical is very variable, and so what I think is in the future, if we can use much more sophisticated machine learning programs to quantify, to subcategory all these behaviors, then it give us much more, because you cannot equal animal behavior, no matter whether monkey or mice, to human. So what we try to understand, if there is a very specific robust behavior, then it will help us to go, okay, what is the underlying mechanism? Then we can figure out how to solve that problem. Right now, to me, seems like the variability is a major issue, right? So that's why we focused on ADHD-like behavior and sleep disorder. These are much more quantifiable, at least for now, from human and from animal. So something that can be translatable between the animal model and the human, That will be very, very helpful for us, for basic scientists or translational neuroscientists.
Thank you. Last but not least, I was struck by an article that I found you quoted in The Atlantic in a recent paper you had in Proceedings that talked about the public and political push to reduce the use of monkeys in scientific research. But your work with Shank3 demonstrates the tremendous potential for genetic engineering in non-human primates to create highly proximate models of mental illness in humans. Can you discuss the promise of genetically modified non-human primates for neuroscience, as well as the ethical issues involved in raising genetically modified higher order mammals?
Yeah, this is a very good question, very sensitive topic. Many of us had discussed this. We recently had several National Academy meetings also discussed this, and we wrote our views. So I have my own views. I think when I did my PhD in Drosophila, and then I graduated originally from medical school. I wanted my research somehow one day benefit the patient. And I moved to mice because I want to get in genetic engineering models and mammals. And at that time I thought I would never go above mice. I did it in Drosophila. Right? But when you talk to patients, you talk to their family, you see their suffering, you really want to help. So then your mind going away is not about you, it's about them. What can we do the best to help patients? So forget about you, whether you are sensitive or you are not sensitive. We are not working for us anymore. We are working for these patients, for their families. So I think we should judge by science and what we need. Every model is useful, actually, but every model has limitations. So my view is that if we need to use non-human primate model, and we definitely need, and if that will bring huge benefit or hopes for patients, we should use it. What is the ethics is also extremely important. They are, for example, I give you a very clear example. I was approached by many family members. Kids have Dravet syndrome. If you have seen Dravet syndrome, they have seizures all the time, and after around 1 year old. Imagine your parents. You see your kids have seizure all the time, and then nothing you can do from 1 year old. Think about it, right? Rett syndrome, same thing. Every parent come to talk to at the conference, they cry. They start crying before they can talk. So, but the mouse model have some good aspect, but they want us to make monkey model because they test many drugs and none of them translate in clinics. I want to help, but I said, if I know this monkey will have seizure all the time, I don't have drug to control it. I just cannot do that yet. I need to talk to my vet. I need a vet tell me, yes, we can control it. We can use the medicine to control it, and then we can study the mechanism. So there are aspects we really have to put front, that we tell the public, yes, there are potential issues, but we do our best to control. But our goal is not working for us, not for any of us to advance our career. Is for each of us to help patients. I think the public will support, will understand.
Thank you so much, Dr. Feng.
Yeah, so thank you so much. Those were great questions, Justin. And there are some questions from those who are listening in, and I can read them. There's one question from Tija Jacob. Are the mice in F1 monkeys raised by Shank3 mutant parents or wild type fostered?
Yeah, that's a very good question. So we did a lot of study look at heterozygous in mice. So they are in mice we raised by heterozygous. We don't foster them, but when they are grow up, they actually with weaned, we separated into genotypes. We found that actually, if you group them together, it's not a wild type monkey mice will make the mutant mice behavior normally. Actually, the mutant mice will make them wild type mice more anxious behavior like a mutant. So we separated after their weaning. They are raised by they never raised by homozygous, and we raised by heterozygous, and we don't foster them. And that's a very important question. For monkeys, we don't fast them, because fast will change their behavior. So they're also raised by the heterozygous mom.
Very good. Thank you. There's another question from Sarah Springer. In the sense
I'm sorry. That actually I said it wrong for the monkeys. So the monkeys, we use the male to mate with the females. Actually raised by the females are Y type. Sorry, the mice are raised by the heterozygous, and the monkeys are actually raised by Y type females.
Okay, thank you. So this next question is, in the sensory hypersensitivity experiments, let's see, in the sensory hyperactivity experiments, did you also try to specifically rescue Shank3 in excitatory neurons and examine the behavioral impact?
In the sensory, in the sensory experiment, we actually knock out we didn't do that exact experiment. We actually did not put the Shank3 into the primate neuron to see whether it can rescue sensory hypersensory. That's a very good question. We did a knockout in excited neuron, and we didn't see a similar phenotype.
Okay, thank you. Let's see, another question. Have you observed behavioral phenotypes in monkeys that are relevant to human patients, but you could not model in Shank3 mutant mice?
That's a really good question. So yes. So the most difficult part, and both my lab and many other labs, especially Huda's lab, I talked to her at the Southwestern Conference Symposium, is the cognitive deficit. You know, in the patient heterozygous, right? Their IQ is around 30, very low. In mice, we tried many different things. We couldn't, water maze or other things. They are very subtle, even in homozygous. So that's the aspect we have been exploring. In the monkeys, in heterozygous, actually our initial, we do see very significant defect in their learning ability, in cognitive function. So we are now develop a touch screen. They actually Y-type learn very quickly. You give them a touch screen iPad, they will, recognize, Nan knows very well. They will recognize shapes, colors, all these different things, and touch 1, touch 2, get different things. So they learn very quickly. So we are now our initial study using Wisconsin, card sorting kind of thing. We did the initial study. We only had, 3 heterozygous. We see some defect, but we really cannot make conclusion. Now we're testing about 20 of these F1s, pure heterozygous. This is the aspect I think will benefit us most, the higher cognitive function compared to mice.
Therapy in the monkeys?
Yeah, so we just is going to start. Now we actually we are trying to determine what is the most efficient route for administration of AAV. It turned out it probably the ICV injections, cerebral ventricular injections. So that's what we'll be doing in humans actually.
Okay, I'm not sure. I don't see any. I wonder if there are any other questions. We have about 2 minutes left for anyone who might wish to ask another question.
If not, I have another question I'd like to follow up on. I was totally intrigued by the developmental time course for the rescue in relation to the anxiety versus some of the other repetitive behaviors. And just wanted to get your thoughts about how you think about that more generally in relation to anxiety phenotypes. Do you think that this is just specific to Shank3, or do you think that this developmental course might be, highly relevant to other anxiety phenotypes?
Yeah, that's a very good question. So actually, it's not specific to Shank3, at least in mice. People have done in Fragile X. They also cannot rescue in adult or later on anxiety phenotype. They also did in SYNGAP1. They also, some of can be rescued in other phenotype, but not anxiety. So I've tried to convince people in the lab to figure out what is the change in the brain that anxiety phenotype you cannot rescue. In many cases, it's not Shank3 specific. So it's a very interesting. It probably have some implication in humans. Early child event actually will cause a long-lasting effect on the kids. So it's a really interesting topic, actually. Very good.
Yeah, that's very, very interesting.
Well, Guoping, I think we're out of time, but this was a great beginning to this symposium. We really appreciate your presentation, and hope you can join us later this afternoon for our general discussion.
I will. Thank you both.
Yeah, just terrific. Thank you so much. Yes. So I guess we're gonna break now and come
we have
back.
A 15-minute break.
We'll come back and then we'll hear from Tom Cash,
Great.
which we're very much looking forward to.
Thank you all.
All right, take care.
Thank you.